Good day, everyone. We are Group 9 presenting CRISPR Cas-Based Prenatal Screening for Aneuploidy, Challenges and Opportunities for Early Diagnosis. For a brief overview of CRISPR, CRISPR means clustered regularly interspaced short palindromic repeats. It is a technology that research scientists use to selectively modify the DNA of living organisms, as said in National Human Genome Research Institute in 2025. Or in simpler terms, it targets specific DNA sequences. As said by Dr. Chan in Cleveland Clinic, CRISPR contains spacers, sequences of DNA left over from unfriendly viruses or other entities, as well as repeating sections of genetic material. Those sequences provide acquired immunity and form the building blocks of the gene editing system or process. It creates a sort of blueprint that allows enzymes in genetic material to make changes to sequences of DNA in living cells. One of the best known enzymes used for this purpose is called Cas9, which is why you'll sometimes hear people talk about CRISPR-Cas9. In this presentation, we'll go over two types of CRISPR, CRISPR-Cas9 and CRISPR-12. So first, CRISPR-Cas9, how does it work? So guide RNA, scientists use uh, or design a guide RNA molecule that matches the DNA sequence they want to edit, and the guide RNA is paired with Cas9 protein, which acts like a molecular scissor. The guide RNA then directs a Cas9 enzyme to the specific DNA sequence in the genome, and once a Cas9 enzyme reaches the target sequence, it cuts the DNA strands. Then, the cell's natural DNA repair mechanisms kick in. Researchers can either influence how the cell repairs be cut, potentially inactivating the gene, or introduce a new DNA sequence to be incorporated during the repair process. It is shown in the picture in this slide how it works. Now we'll go over CRISPR-Cas12a. Cas12a, guided by the cRNA, performs targeted DNA cutting, and once activated, non-specific single-stranded DNA cutting is performed. Unlike Cas9, Cas12a processes its own cRNA, recognizes time-enriched protospacer adjacent motif sequences or PAM sequences, and it cuts farther from the PAM, enabling multiple edits. This is expressed in engineered bacteria using vectors like PET15B or PET28A, then purified through cell lysis, chromatography, STS page dialysis, and Western blotting. Genome sequences from databases are analyzed to select specific target size and design primers or probes and verify specificity to avoid cross reactions. Clinical samples are collected, nucleic acids are extracted and amplified, and then incubated with Cas12a cRNA and a fluorescent probe to detect target DNA via fluorescence signals. So, the historical timeline of CRISPR begins in 1987 when Ishino and his colleagues first observed unusual repeating sequences in the DNA of E. coli. At the time, no one knew what they were for. So, from 1993 to 2005, Francisco Mojica discovered that these repeats were found in many microbes and noticed that they matched DNA from viruses, hinting at an immune defense system. In 2005, Alexander Bolton identified the Cas9 protein in Streptococcus thermophilus and described the protospacer adjacent motif or PAM as a critical signal for recognizing target DNA. So between 2006 and 2007, Eugene Koonin proposed the immune function of CRISPR, while Philippe Horvath proved it experimentally by showing bacteria could acquire viral DNA and use it to defend against future attacks. So from 2008 to 2011, researchers revealed how CRISPR works. John van der Oost showed that viral DNA fragments are transcribed into guide RNAs. Marafani and Sontheimer confirmed CRISPR targets DNA rather than RNA. Sylvain Monod discovered Cas9 makes a precise cut near the PAM. Emmanuel Charpentier found track RNA, which helps guide Cas9, and Virginus Sixnis demonstrated CRISPR works beyond its original bacterial species. Then, in 2012, Charpentier and Jennifer Doudna simplified the system by combining two RNAs into a single guide RNA, while Silkness characterized Cas9's cutting mechanism. And finally, in 2013, Feng Zhang and George Church adapted CRISPR-Cas9 for use in human and mouse cells, making the start of a new era in genome editing.
CRISPR has shown potential in advancing prenatal screening for aneuploidy, a condition where an embryo or fetus has an abnormal number of chromosomes. Common aneuploidy conditions such as Down syndrome, Edwards syndrome, and Patau syndrome are typically screened for during pregnancy. This is currently done via non-invasive prenatal testing, or NIPT, or invasive methods like amniocentesis. However, the accuracy of NIPT relies on a high number of cell-free fetal DNA, or CFF DNA, in the mother's blood, while invasive methods can potentially cause physical harm to both mother and the fetus. With CRISPR, regions of fetal DNA in the mother's blood can be selectively targeted and amplified to signify chromosomal abnormalities. This allows for prenatal screening with earlier detection, higher accuracy, and reduced risk compared to traditional methods. In a study by Ganesh et al., they discussed the potential advantages in the application of CRISPR to prenatal screening. In it, they cite several studies that showcase the use of CRISPR to detect non-infectious diseases, as well as its use in editing and diagnostics of prenatal screening. You can view them in the following tables. As with all studies, the advantages that CRISPR provides must outweigh its potential hazards to be considered ethically sound. The main concern is that ease of access to safer screening may cause an increase in the number of terminations for fetuses with chromosomal abnormalities. That might not be the case, though, as a study revealed that while only 8% of those diagnosed with Down syndrome using invasive methods were carried to pregnancy, this number increased to 31% for those using NIPT. This may even increase if a more accurate screening method is introduced, like CRISPR. Nevertheless, this application may enable healthcare professionals to recognize the true capabilities of CRISPR.
With the advent of the CRISPR-Cas9 system, genetic diagnostics have been advancing through the years. Ganesh et al. Study details how CRISPR can be utilized in non-invasive prenatal screening for aneuploidy. First, plasma samples are collected from the maternal blood. The cell-free fetal DNA, or CFF DNA, is then isolated from the samples using standard PCR kits. The CFF DNA is then amplified using fluorophore-labeled primers designed for recombinase polymerase application, or RPA, which contains single-guide RNAs, or sgRNA, and Cas nucleases that target chromosomal regions to detect aneuploidies. The sgRNAs are also programmed to target trisomies such as trisomy 13, trisomy 18, and trisomy 21. The sgRNAs target and bind to the fluorophore-targeted CFF DNA, forming a complex, and then the fluorescence generated by the separation of the fluorophore and the quencher is measured using a spectrofluorometer with distinct absorbance and emission wavelengths. Despite CRISPR being revolutionary, they still come with limitations. The primary challenge of CRISPR-Cas-based diagnostics is the development of reliable and accurate gRNAs, or guide RNAs. They must be specifically programmed to target specific chromosomal regions, which require a complex understanding of the genomic areas and are unique to each chromosome. There's also the possibility of binding to non-target genomic regions, which leads to false positives or false negatives. Cas9 and gRNAs are also large and negatively charged, posing a challenge when entering cells due to the cell membrane. While various methods to deliver CRISPR, such as plasmid DNA, exist, each method comes with its own necessities and problems. Lastly, targeting specific tissue or organ types also requires tailored delivery methods, as what works for one may not work for another.
Ongoing research into CRISPR-Cas-based prenatal screening for aneuploidy is moving at an extraordinary pace. Today, I would want to walk you through some of the most promising breakthroughs shaping the future of early genetic diagnosis. So, a remarkable milestone in 2024 where Liu and his colleagues showed CRISPR-Cas9 could selectively remove an extra chromosome 21 in humans-induced pluripotent stem cell and fibroblasts. This process is known as allele-specific chromosome elimination, and this successfully normalized gene expression pattern in vitro. While it's still at the lab stage, this opened doors for possibility of future therapeutic intervention for trisomy condition. On the diagnostic side, the CRISPR toolkit expanding rapidly. In 2019, Kellner and team, and later Meyerbold in 2022 adopted Sherlock, Cas12, and Cas13 platforms to detect extremely small amounts of nucleic acid in plasma. The result? Rapid, inexpensive, and potentially point-of-care prenatal test that could be used even in resource-limited setting. Another major leap come from Chen and colleagues in 2018 and Wang's group in 2021 that allowed combined isothermal amplification of Cas12 detection. And this is highly sensitive analysis for minimal sample volumes, which is ideal to detect fetal cell-free DNA circulating in maternal blood. And in 2021, Faustina's research took a step further by creating miniature multiplex Cas12 sensor array. This can detect multiple genetic targets simultaneously, making it possible to distinguish whole chromosome changes from single gene variants into one test. And finally, in 2023, Liu's team have created an integrating fragmentomics. This studies pattern in DNA fragment size, methylation, and nucleosome positioning into CRISPR-based assays. This boosts sensitivity and reduces false positive, enabling earlier and more reliable aneuploidy detection. Now, moving into the healthcare implication, we know that CRISPR-Cas has a huge implication on this sector. As Zhang and colleagues noted in 2021, CRISPR-based diagnostic could make prenatal screening faster, cheaper, and accessible worldwide. Multiplex platforms like those demonstrated in Faustina's team could allow screening for multiple conditions, from trisomies, microdeletion, and even some single gene disorders, all at once. Looking further ahead, Liu's 2024 study on trisomy rescue raises the possibility of therapeutic intervention. While we're still far from that reality, this is a major safety delivery and ethical barriers to overcome before in utero application can be possible. Wilson and colleagues in 2020 reminded us with such powerful tool comes the responsibility to ensure rigorous clinical validation, adherence to the guidelines, and strong ethical safeguards. These include informed consent, equitable access, and disability awareness for counseling of families. So, from chromosome editing to portable diagnostic devices, CRISPR revolution is reshaping the frontier of prenatal medicine. The challenge now is not just to push the science forward, but to do so responsibly, equitably, and with the needs of all patients in mind. So that's all for our presentation today. I hope you have learned a thing or two about CRISPR-Cas9 and how it is used in aneuploidy detection and the obstacle and success within it. Thank you so much for listening.